So it's my pleasure to introduce the speaker today, Dr. Chad Cowan from the Harvard Medical School. So Dr. Cowan finished his PhD at UT Southwestern and then moved to Harvard to do his postdoc and has been there for the last 15 years. He's moved from the Harvard College over to the medical school in the last 7 years, and he also directs the Harvard Stem Cell Institute core facility there. Dr. Cowan's research interest has been in studying, has been in using iPSCs to study the effect of genetic variants and their effect on type 2 diabetes and coronary heart disease. He has done some work on the Framingham heart study where he's taken peripheral blood samples from participants in the Framingham heart study, converted them to iPSCs, and then differentiated them into various cell types and identified functional importance of genetic variants that were identified in genome-wide association studies. So welcome.
So, thank you, Barack, for the very kind introduction. It's really my pleasure to be here and to talk to you about some of the research we've done, spanning the last maybe 10 years. I will take you back in time with the first slide or so to what drew me to Harvard and sort of, where we were at conundrum-wise, and then sort of try to do my best to be a salesperson for why we use the models and systems we use to try to understand disease. But I also have a tendency, and I've just given myself over to this rather than fight it, to speak really quickly. And if you have any questions or if you didn't understand something, just stop me and I'll be happy to answer any questions. So when I first came to Harvard, the human pluripotent stem cell had just sort of dawned, that era of, wow, we might be able to do something with human embryos. But I think more importantly, this concept had just been born. This was the concept that really captured my imagination, because once we, sort of started thinking in the most ambitious way about human pluripotent stem cells, which one of their true powers is the ability to become any cell type in the human body, one of the most ambitious ways to think about that is like you're studying a little human in a dish. You have human biology opened up to you in a way that you had never had before. And if you could couple that with something that had happened, decades before, which was this idea of cloning, maybe you could start making stem cells from anyone. That was the concept that really started sparking my imagination when I was at Harvard. And we knew it was possible because of what had happened with John Gurdon's frogs and later with Dolly the sheep, that you could actually take a somatic nucleus from an adult organism, put it back into an egg that had been enucleated, and make the whole organism again. And we didn't need to make the whole organism again. We just wanted to make the embryo from which you would normally derive a pluripotent stem cell line. So this idea that you could make a stem cell line that would represent a particular patient or a particular person was really, really exciting. And it was exciting because at the time I was in a laboratory that was struggling with trying to understand a disease, type 1 diabetes, which has a lot of genetic components, has some environmental components, but like one of these stunning facts, you can have identical twins and one of them will get type 1 diabetes and one of them won't. And what if you could do this fantasy experiment of making the two twins and sort of watching them on the same island and feeding them the same food and then seeing, that's the one that got it. That's sort of what this lets you do maybe in a dish. Of course, when we entered the fray, all of this was still quite messy. Like, it was conceptually true that you might be able to do this, but it was far from easy. And in fact, the way in which we first conceived of it through nuclear transfer turned out to be the longest path to success. Our contribution to sort of what happened here was we were the first to sort of show that if you took a pluripotent cell and fused it to a somatic cell, you could actually get something at the end that thought it was pluripotent. It thought it was a pluripotent cell. The only problem was it had the nuclei or the genetic material from both cells, so that was a mistake. But what it taught us was that there was everything you needed in a pluripotent cell to become a pluripotent cell. So then scientists are really good at this. Then it's a reductive approach at what is the stuff, what's the magic, witch's brew in a pluripotent cell that can make a cell become pluripotent again. And that's where the hunt was when Shinya Yamanaka came along with a stunningly simple method for reprogramming somatic cells like skin fibroblasts, and in his case he used a tail tip fibroblast from a mouse, and just 4 transcription factors, which are typically found in embryonic stem cells, to make something that he termed an induced pluripotent stem cell. In other words, he could almost make them like we used to do with transcription factors that cause cancers like oncogenes. You could make it catch pluripotency. You could basically give it this little special property. And so this made possible for the first time in a real sense the ability to do this. Identify patients or whatever people you wanted to, reprogram with these defined factors to a patient-specific pluripotent stem cell. And I'm going to speak today entirely about what we've done in trying to understand patient diseases and patient phenotypes in an in vitro model. My lab has spent the last 7 years trying to understand how we might push the envelope towards cell-based therapies, and if you want to talk to me about that later, I can talk to you about it, but it's just too much to put into one. And I was originally born in Kansas. That's where I grew up. I think I told my host today that the last time I was at the University of Minnesota was when I was swimming for the University of Kansas. I think that's the last time I was here. But one of the things that is different about Boston and Kansas is in Kansas you don't really walk anywhere, you don't run anywhere, you drive everywhere. And as a result, this epidemic of metabolic disease is rampant there. And so when I set my sights on what I wanted to understand as a scientist, I set my sights on understanding obesity and what really drove obesity. And I thought, one powerful way to use this would be if we could take obese patients and start to understand that metabolic syndrome with this in vitro model, this might be relatively powerful. And why was I so taken with obesity? It's because it's an incredibly rampant problem. And when I talk about this in Boston, it's all these, healthy young people that are there and they just don't see it. And I go home to Kansas and I see my poor parents and I see everybody else really devastated by the disease. Easily 30% of the people in the US are considered obese. That's like with a BMI over 30. I think what's most stunning to me is as countries get increasingly economically stable and adopt Western diets, you see this sort of rampant throughout the world now. It's the largest problem in China, and it's starting to happen in India. And what's surprising to me is in both of those countries, with lower BMIs, you're getting some of the same metabolic complications. So it doesn't take as much, it seems, to really push the needle across. And of course, there's major health consequences. I mean, being fat and obese is one thing, but because of that obesity and the consequential metabolic problems, you almost always get diabetes. So obese people typically get diabetes, and those people with diabetes typically die of heart attacks. And we wanted to understand, how does all of that happen? Of course, the problem with metabolic disease is it's really, really complicated. It's not just a disease of one cell. It's a disease of many of the cells in your body that try to handle energy deposition and energy usage talking to one another. And it's this complex interplay between all of these cells that typically leads to what we often call metabolic disease, metabolic syndrome, and then eventually the other things I just talked about. Add to this the fact that we know that there's genetic risks, environmental risks, behavioral contributions, and it becomes such a mess that it's really difficult, at least for me as a simple PhD, to tease apart the pieces of the puzzle. That was why I thought that this idea of simplifying it, like let's just take the genetic components, put them in a cell and say, what's the genetic contribution to aspects of this versus what are the environmental things, what are the behavioral things, like what are the things that are putting people at risk for the disease aspects that I'm interested in. And at the time, I became really fascinated with fat tissue, and that's because in this metabolic disease, it's the thing that changes the most dramatically that you can see. Sometimes it takes up to 70% of a person's body weight. That's stunning to me. It's the only adult tissue that's known to sort of shift in its mass to that degree once you've actually achieved your full growth and body size. So I was really taken with the idea of like, something about this is directly contributing to metabolic disease, and what is it? What's the link between fat, the metabolic syndrome, and diabetes and heart attack? So we started studying fat, and this cartoon is to illustrate everything people know about fat and how it contributes to it. And it's an oversimplification, but I kid you not, this really is approximately what we think is happening. You have normal adipose tissue, you eat too much, you have an excess in nutrients. That excess in nutrients expands the adipose tissue to a point where it's now stressed. It's stressed out. This is beyond what it can normally do. It starts sending a lot of stress signals and signals to the immune system, because for it to remodel, it needs the immune system to do that, to grow, etc. And more importantly, because it's gotten so big, it actually starts to become oxygen deprived, etc. And you've got a tissue that now is now thought of as dysfunctional. I like this simplified cartoon because when you go to a review, you get this much less simplified cartoon that says the same thing. This same cartoon says this is like a disaster that happens because of dysfunctional fat, because it's taking in excess nutrients. And the only thing the reason I add this more complicated cartoon is what it reminds you is that fat talks to everything else in your body. It's talking to every other organ, and it's talking to your brain. And so what's happening to your fat cells is also sort of being communicated and relayed to the rest of the body. So that's the introduction. Now I can tell you, like, how did we think about approaching this? How did we want to tease it apart? And I stole this shamelessly from David Altshuler, which he has this bridge of how do you fill in these clinical therapies. But I think it's right. So I'm starting with what are fundamental biological processes, and I've always wanted to know, like, I won't feel satisfied as a researcher unless I've made a difference for a patient. So I want to know, how could we get to something that might actually be a meaningful clinical therapy? And this is an enormous gap in biology, in fact. So there are many ways to sort of, fill in that gap, but we've thought of one method that we've been sort of now using, almost just turning the crank on, and so I'll sort of walk through it. The first step, in our opinion, is to try to understand as much as you can from the naturally occurring experiments in humankind. That, all of us have a different predisposition towards obesity, diabetes, and heart attack, and it's only in the last 10 years that we've really started to explore deeply on a molecular level what that is, and that's usually using genetics. But even before the last 10 years, people have done these really beautiful epidemiological studies which have given us a lot of information about the things that lead to these diseases. But using that, we can basically say, here's what we think are the genetic contributions, and then once you have a genetic contribution, you want to do this really simple experiment of, like I said, have identical twins, but sort of add or take out a gene, and then say, in this twin that has the sort of disease-causing gene or mutation, what happens? And that's what we try to do with stem cells. So we use stem cells, gene editing, and the ability to turn the cells into any of the tissues that are affected in metabolic disease to try to explore those things in vitro. And if we're successful, we can pat ourselves on the back and say, we now have a relevant way to understand that disease and its phenotype in a dish, which we call a disease model. And those disease models are really powerful ways to start to explore how to fix that biology if it's gone wrong. And one of the ways you can fix it is with a small molecule, and we've done these, and I'll show you some evidence where you can do a drug screen on these models. And then if you're really lucky and things have worked out, you can enter into an industry partnership. I no longer believe that I'm going to be a PhD in a hospital, like, delivering the drug. There's just absolutely no way. The way in which drugs really truly get made is through a partnership with biotech and industry, which, sort of commercialize it and make sure that it's safe. And I try to tell students these days that that's what you want to, if you've achieved the right thing, you've learned enough about a system where you know that this is the target, and maybe you've got the initial leads on how to drug that target, but you're probably never going to go beyond that, nor should you try in an academic setting. Okay, so the way I've structured my talk is the way Sidney Brenner says science is structured, and I totally agree with him. He says that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Students find this completely controversial, but it's almost certainly true. You'll have an idea like, ooh, I want to answer this question, but you can't answer it because you don't have the technology to sort of dissect it, to approach it. So it's almost always these technological leaps that now give you access to things that then lead to new ideas, and then, of course, the new discoveries. So I think that this is a way to say that we're going to talk about some of the things that we've done in terms of technology, then the discoveries, and a little bit of future directions. So we've had to spend 10 years building tools, which is really frustrating at the end of the day, but it does open up avenues that you would have never done. And the two sort of tools that we've had to do a lot of work on is the ability to turn these pluripotent cells into an adult cell type that you can reliably use in a laboratory setting to understand something about disease. And then I think another important thing is, if you want to understand genetics, you sort of want to rewrite the genetic code in a way that would let you say, let's look at the disease state versus the non-disease state. And so we've really had to play with some of the genome editing tools. So what was our goal? Our first goal was to try to make some fat in a dish. And I remember when I was trying to tell my mother, I always feel like you're not a successful scientist until you can tell your mom what you're doing. And I'm trying to tell her at Thanksgiving once, Mom, I'm trying to make fat. And she's like, Chet, I don't mean to tell you your business, but maybe you've got this backwards. You're trying to cure obesity. That means not making fat, right? And I was like, I know, Mom, I know. But making a lot of fat is one of the keys to understanding this. And so she was like, okay, sure thing, Chad. But hopefully I'll be able to convince you that making a lot of fat routinely, robustly, and sort of the same way does unlock a lot in the lab. But when we first set out on this, and I guess it's now over 10 years ago, it seemed really simple. Like I cracked open a textbook, I saw one of these cartoons, and you know, in most of the world of differentiating stem cells, what we try to do is we just rip a page from what we understand in normal biology and normal development, and we use that as the playbook for what we want to try to do again in a dish. And when I looked at this review, it was like, oh, I've got the playbook. I got to take a pluripotent cell, I'll turn it into this mesenchymal progenitor, and then a pre-adipocyte, an immature, you know, and all the way to an adipocyte. And I was like, this is going to be a piece of cake. The problem with this cartoon is it's science fiction. None of the developmental biology has actually been done that explain the steps, except for maybe the, you know, early mesenchyme. But the specific mesenchyme that gives rise to different fat tissues isn't well understood. Those signals that then commit it to a pre-adipocytes fate, on and on and on. Completely science fiction. It's just happening now, but it's been one of those areas that has been underexplored. So I couldn't steal that page from developmental biologists that would allow me to recreate it in a dish. So instead I had to take a page from Yamanaka's playbook, which is what he said, you know, like if you can understand the transcriptional master regulators in a cell, you might be able to, like you can give a cell cancer, make it, or catch pluripotency, maybe you could make it catch fat. So luckily, the work of Bruce Spiegelman and others had, over the years, essentially described that transcriptional hierarchy that made a fat cell a fat cell. And in fact, one of the key, you know, first master regulators discovered was this gene PPR gamma, which is really known to be essential in fat, and not just essential, but capable of making fat. So we had a really simple idea, which was, what if we just sort of gave the cells this PPR gamma in an inducible way? In other words, we could turn it on or off whenever we wanted. Would they become fat as a result of that sort of initiating the transcriptional cascade that's normally in fat cells? And while it was simple, I can tell you when I had this idea, everybody bet against it. You know, so what's funny is after it works, everybody's like, that's just obvious. But when you first do it and you haven't done it, everybody's like, there's no way that's going to work. Like, there's 99 reasons why it shouldn't work. So then it works. You put in PPR gamma into these mesenchymal progenitors that we can make relatively easy, and 95% of them become something that looks like a mature fat cell. That's in contrast to when we throw everything we know at them about sort of differentiating fat cells, you know, like try to make them fat with every sort of known cocktail where we can maybe get 5 to 10%, and they're always really, really immature. And we did this work now, I'm ashamed to say, like, or at least published it over 6 years ago. And once we'd done that, it sort of unlocked the key to doing it with almost any cell. And you're seeing a lot of groups do this now, where they might use a transcription factor to sort of just jumpstart that differentiation protocol, because it's often faster and more efficient than trying to add growth factors or other factors that are going to initiate the same transcriptional cascade at the end of the day. So sometimes they just go skip the middleman and go straight to the transcription factor. This works not just for the white fat that's involved in most metabolic disease, but this other really interesting fat that people are, you know, now becoming more and more interested in, which is brown fat. You can add the transcription factors that we think are essential for that and get something that is able to store lipid, but rather than just store lipid like fat does, it actually uses that lipid to produce heat. That's what brown fat does in babies, and if you're lucky enough to have some as an adult, that's what happens if you get cold in a room before you start shivering. In non-shivering thermogenesis, this stuff basically burns that energy and creates heat. And it's known to be really beneficial. The more of that you have, the less of your risk of a lot of the metabolic complications I've talked about.
So what did you do to make the brown?
Just added different transcription factors, and in fact, I put it up here because it's controversial in Boston. PRDM16, Bruce Spiegelman had defined and talked about. It's completely dispensable. It's in fact not even involved in brown fat. It's in fact in subcutaneous fat. But the only factor you need is CBP beta seems to be the critical difference in terms of a transcriptional regulator. It starts the mitochondrial program that is essential for a brown fat cell. And the PPR gamma sort of makes you fat-like. So those two together make things that are indistinguishable from brown fat in our hands, so indistinguishable that we've actually transplanted these cells into mice, immune-compromised mice, and they're functional. You can rescue immune-compromised mice that aren't able to make fat, so these little skinny immune-compromised mice we've made, you can put fat, white fat in them and it rescues them, and you can put brown fat in them and it's functional. You can do the PET CT scans that allow you to identify it. It's energetic and everything else. So this is just a long summary. If you care about it, the papers are already there, but the point is we can make fat. Okay, now that we make fat, what we want to do is be able to introduce disease-causing mutations that we've identified in people in fat or in stem cells, which at the time that we wanted to do this was really, really hard. It was like when I first started my PhD and you wanted to make a knockout mouse. You would basically create this gigantic construct and try to do homologous recombination, and like 1 in 1,000 or 1 in 1,000,000 would actually happen, and it was unbelievably difficult and laborious. Luckily for us, these tools that allowed you to more precisely edit the genome, which is to make any sort of cut you want with these engineered nucleases, came along. And when I started, we were using the earliest of these engineered nucleases, which are meganucleases, but then zinc finger nucleases. And then sort of a godsend was another class of them called talons. And then eventually, I think everybody in the audience now knows the CRISPR-Cas system. Whichever of these engineered nucleases you use, what you're ultimately doing is creating a double-strand break. That double-strand break can either, here's what people always forget, after you create the double-strand break, you basically let the cell do the work, right? The cell's actually doing the rest of the job for you. And cells basically heal double-strand breaks in 2 ways. 90 to 99% of the time, it's through non-homologous end joining, where they just grab the 2 free ends and stick them together. It's not perfect, and so occasionally you'll get deletions or insertions, and that's good ways to disrupt genes. And the other way is what we call homology-directed repair, which is typically only active during the cell cycle. And it's when after the break happens, it basically says, wait a minute, we have another copy, it's nearby, looks at the sister chromatid as a template, and then just copies that in. If you supply stuff, much like Oliver Smithies and Mary Kopecky did, that looks like the sister chromatid, you can do homologous recombination, and that's how you make precise changes. Okay, so I'm going to bring you full circle now. Now I have the tools. I can make fat. I can make cell types involved in metabolic disease. I can make genetic changes. I can try to do the experiment I talked about at the beginning, that twin experiment, where I basically take a normal person, introduce a gene mutation that I think is involved in a disease, take these 2 identical twins, and in this case it's identical twin cell lines. People often call them isogenic in that they're identically genetically except for the mutation I made, differentiate them and make these very precise phenotypic comparisons. And that's what I'm going to tell you about sort of for the rest of the talk, is how we've used these tools and some others, which I'm not going to go into as much detail, to dissect a lot of disease mechanisms and understand more. So one of the first things we did was try to understand some of the key regulators in insulin signaling, because if you know anything about diabetes, the thing that is the hallmark for diabetes is insulin resistance, and insulin resistance is defects in insulin signaling at some level in the tissues. And in the insulin signaling pathway, again, I have an uber simplified cartoon. It's one of these things that has like hundreds of factors that influence it. But what we know is that insulin talks to the insulin receptor. The insulin receptor uses these key downstream kinases that are intracellular mediators of that signal, AKT1, 2, and 3. 2 is the main player in metabolic tissues like liver and fat. AKT2 phosphorylates the transcription factor FOXO1 and brings it out of the nucleus, thereby changing gene transcription in response to insulin. So that's a very simplified way to look at it, but it sort of highlights how critical this one player AKT2 is. And what happens when you see insulin as a fat cell is you increase glucose transport. This makes sense. You've just eaten. You've gotten an insulin bolus. You've got a bunch of glucose in your blood. You're going to increase the amount of glucose fat cells take up, and then they turn that glucose into lipids. They'll also take up lipids that you've eaten, dietary lipids, and store those. So they're essentially the reserve gas tank for fuel. And the problem is most of us are consuming more fuel than we're burning, so you fill the tank up and keep filling it. Okay, so why was I interested in AKT2? The reason I had become interested in it is in 2011, a friend of mine, Robert Semple in the UK, had identified this very interesting family that had a mutation in AKT2 that actually led to hypoglycemia, obesity, and left side overgrowth. And it was a missense mutation that took a glutamate and made it into a lysine at the 17th position. And why did that seem like such a fascinating mutation? The reason why is it was the first of its kind in that we already had loss of function mutations in AKT2, and they had the opposite phenotype. So when you lose function in AKT2, you're hyperglycemic and you have decreased body fat. You're lipodystrophic. So we know that if you lose function, you probably are ablating insulin signaling, whereas this, for the first time, looked like a gain-of-function mutation, the first of its kind in the insulin signaling pathway. So we were like, maybe it is. I'm really interested in it. And I contacted Robert, and I was like, Robert, let's get those patients. Let's make iPS cells. 5 years later, still haven't even done anything close to that. But luckily, we had genome editing tools, so I could just introduce the mutations. So that's what we did. We knocked out AKT2, just like some patients with lipodystrophy. We introduced the E17K mutation, and it doesn't do anything to the protein levels, but you can see that you now have a lysine. And we could ask, what happens when you do that? Well, we had the first evidence that it's a true gain of function. So when we looked in fat cells, what I'm staining here is FOXO1, that transcription factor, normally resident in the nucleus of cells unless they receive an insulin signal. This is what happens in a normal cell, resident in the nucleus. Upon receiving insulin, it's redistributed to the cytosol. If you're a knockout, you don't respond at all. Doesn't matter how much insulin I dump on you. And if you're the E17K, it doesn't matter if I've stimulated you or not. Most of the FOXO1's out in the cytosol. So it's a gain-of-function mutation. What's that mean if you're a fat cell? It means that you're taking up a lot more glucose. In fact, the E17Ks are off the charts. They're beyond my insulin-stimulated glucose uptake in a wild-type cell. They're always doing it. Because they're always doing it, they get fat. This sort of explains the obesity you're seeing in the patients. And the other fat cells that are knockouts are lean. So we have the first sort of molecular real understanding. The surprise, though, came in the following. When you turn on insulin signaling constitutively in fat cells, they do something that I never thought that they would do. They send out stress signals. And what's interesting about that is there's a lot of ways in which you can grow fat cells, make them bigger, et cetera. It seems to be intimately linked to the insulin signaling and the constitutive insulin signaling that leads to this inflammatory cascade. So the fat cells themselves are sending out inflammatory signals, IL-8, MCP1, et cetera, which are known to draw in macrophages. Those macrophages then basically amplify that signal, and you get that initial inflammatory state, which we know is critical to the insulin resistance and pre-diabetes that establishes in people. And there's been years, if not decades, of debate about what's the chicken and the egg in this inflammatory response. And I think a lot of the community was sort of pointing its finger at the immune system as the bad guy, like something's gone wrong in your immune system and that's what's doing it, and it's dysregulation there. I can tell you that the genetics for diabetes, the genetics for obesity, the genetics for insulin resistance say nothing about the immune system. There's not a single immune-specific gene that's come up. It's almost certainly something else. You can say that the immune system is working perfectly to respond to what is probably other tissue dysfunction. And I would argue that probably the principal tissue dysfunction is the fat cell. We still don't understand the molecular basis of how misregulated insulin signaling is changing the inflammatory cascade, but we're doing more work on this. And what's interesting to me, I don't have it in today's talk, is no matter which way you turn it on or turn it off, if you dysregulate between like what would be a pretty narrow therapeutic index is the way I think of it, you're going to get problems in fat either way, and either way sends signals to the tissues in the rest of your body. Okay, so what did we learn from just this study? We learned that we could understand the molecular basis of the phenotypes in this patient, but I think most interesting to me that adipocytes are the principal players in this initial inflammatory cascade that leads to prediabetes and diabetes, and that's been a big subject of research in the lab that we still haven't figured out. So let's just change subjects, since I don't know more about it, to something else. Bharat mentioned this at the beginning, which was we chose to try to do something people hadn't done in cells yet, because what you really want to do is not understand this really, really rare patient with a monogenic disorder. What you want to do is the people that come to the hospital every day and have metabolic disease understand something about that. And so we sort of pushed ourselves to ask the question, could we use iPS cells to understand common genetic diseases, these things with really subtle, mild effects? The problem there is it's a bigger issue. So the way in which you do the genetics of these common genetic diseases is you get thousands of people, and in fact hundreds of thousands of people. In this particular study, we've most recently published one which is 300,000 individuals. And I always joke, because say Katharayson, who I've done most of this work with, will say that this is a high school student could design these studies. In essence, you take patients, and when it comes to heart attack, the patients are people who have had a heart attack before the age of 40. We call that sort of heart attack genetic risk, because then you can sort of minimize environmental, behavioral, and other things. It's almost certainly like you got a bad draw on the cards and you had a really strong genetic risk to get a heart attack that early. And the non-patients are people who haven't had heart attacks, and especially if they're older than 40 and still haven't had a heart attack. And you basically look at the DNA in these individuals, and you ask, is there any statistically significant DNA difference that's found in the patients, which we now call a risk factor, or something found in the non-patients, which we might call a protective factor. And we've done this over and over again, and we keep finding more and more and more genes. And one of the genes we found when we first did this was this, what I should say is the first thing we learned was genetics works. Genetics taught us everything Brown and Goldstein had taught us over 40 years about what are the main risk factors for heart attack. It turns out it's lipids, and in fact LDL cholesterol. So the who's who of LDL cholesterol showed up when we did this genetic study of heart attack and lipids. And in fact, you've got the target of statin here. You've got the LDL receptor, which is what led Brown and Goldstein on their merry chase to begin with. you've got ApoB, you've got basically a who's who in lipid biology. But the top hit was one of these things that didn't have a gene associated with it. Brown and Goldstein hadn't taught us about it, and it was the thing that seemed to be the most significant risk factor. And when you talk about common genetic disease, we're talking about fractions of a change over your lifetime. This is a much bigger fraction of a change. If you were one of these lucky devils who was a homozygous minor, you could actually expect to have a 16 milligrams per deciliter lower LDL cholesterol. To put that in context, that's like taking a weak statin for your whole life. You know, so you have a 40% reduced risk of coronary artery disease if you're that lucky devil, which is a pretty huge change when we talk about common genetics. And unfortunately, we knew nothing about it. So then begins the chase. What do we do? So when we looked at where is that genetic change, what we found was it was in a really busy part of the genome that's in linkage disequilibrium. All these genes sort of travel in heredity together. But unluckily, and this is true of 95% to 99%, it depends on which genetic study you look at, of these genetic changes, it wasn't in a gene. It was in fact in between some genes in a non-coding region. So everybody at this point in genetics was scratching their head and saying like, well, how the heck do we chase these down? Sort of the most easy sense to think about these genetic changes is what we now know about the non-coding genome is it regulates the coding genome. So we started to wonder, could this affect the regulation of nearby genes? And so we did one of the first of its kinds, one of these expression studies where you look at the genotype versus gene expression in tissues that you think are important for instance, lipid biology. So we teamed up with Lee Kaplan, and he's at MGH, and when he would go have patients come for the bariatric surgeries, they'd take little pieces of their fat tissue, little pieces of their liver tissue, and also a little piece of skin that we didn't even talk about, and then we could do expression analysis and genotype. And what was cool about that is we can now do this thing where we could take my homozygous major genotype or minor genotype and ask, does gene expression change in any of those tissues when we've done that study? In the fat, nothing changes. We don't see any genotype-specific expression change at this locus, but in the liver, 3 different genes change in specific with the minor allele where we know there's some sort of effect here. And I've highlighted one of them, sort 1, and I've highlighted it because I'm just going to make a really long story short, which is we figured out that sort 1 was the principal player in this. This gene, sortilin, which is really well named, sorts things at the ER to either the lysosomal destruction or to the surface. And so if you have higher expression of it, you actually get lower levels. Higher sort 1 gets lower LDL. So those lucky devils with the MM actually have the following thing happen. That single nucleotide polymorphism creates a transcription factor binding site. That transcription factor in the liver turns on sortilin expression at higher levels than you see in other people. Higher sortilin levels reduces the amount of LDL. And we've done 2 or 3 studies on the molecular basis of that, and you're welcome to follow up on it, but that's the idea. So now the question was, we spent 7 or 8 years tracking all this down in mouse models and everything else. Could we have done that just by taking a bunch of patients from the genetics study and found the same sort of thing? Would we have found this study without having to, recruit hundreds and hundreds of patients in bariatric, et cetera? And that was the idea here. Could we use iPS cells to do the same type of study? Now you have to rethink it. You can't start with 1 patient or even a couple patients. You now need to start with a much larger group of patients, a population that's going to give you that sort of insight. And when we did the power calculation on this originally based on the effect size, even though it was strong, we thought it was probably going to be several hundred patients. So that was a little daunting, and luckily the NIH decided they would pay for something like that. So we set out on our merry way. And what we did to get access to patients which had the right information for us to have a good basis to then move forward and do the test was we paired up with this nearby group in Framingham where they're doing the Framingham Heart Study. I'm ashamed to admit it, I didn't know anything about the Framingham Heart Study until I paired up with these guys. This is the best study that's ever been done in the U.S. So right after Franklin Delano Roosevelt basically died of a stroke, the U.S. decided, oh my God, we don't know anything about heart disease. We don't know anything about strokes. We don't know anything about heart attacks. And it doesn't surprise me at all. I've been talking to older cardiologists, and they say 50 years ago, if you had a heart attack, it stuns me. Guess what they would do to you in terms of treatment? This is state-of-the-art treatment. They would put you in a quiet room where it was dark and just see how things went. For 3 weeks. For 3 weeks. 3 weeks you'd be in a dark, quiet room. So it's not a shock to me, nor should it be to you, that 90% of people that had a heart attack succumbed to that heart attack and died. 90%. The quiet room didn't necessarily fix them up.
It was actually up to 6 weeks.
There you go, up to 6 weeks. There you go. So you turn that on its head. turn that on its head. Today, if you get to a hospital after a heart attack within, say, 15 minutes, 90% of you will survive, and you'll survive for greater than 2 years. So our understanding of what causes it, how to treat it, everything has changed. And a lot of that came from the Framingham Heart Study. We didn't even know what led to heart attacks until they initiated this. And the reason they chose Framingham was because it was the most plain vanilla town in all of America. It was an industrial town. Everybody looked the same. Everybody ate the same stuff, same socioeconomics. So they were like, let's just pick these people and kind of follow them along, and we might learn something. Within 10 years, they learned a lot. All the stuff you take for granted, like, oh, that's a risk factor. In fact, they coined the term risk factor in their first publications. The fact that smoking is bad for you, high blood pressure is going to lead to all the sort of standard epidemiological common things that lead to heart attack, they figured it out in 10 years. And I would say the NIH did the smartest thing it's ever done, which it never does this. It doubled down. It actually said, let's put twice as much money behind this study to see what else we can learn. And they tried to do something more interesting with the second group they recruited. In the first group, they just recruited everybody that they could. In the second group, they were like, let's do this a little bit more smart. Let's get people who don't have diseases yet. So a bunch of young, healthy people. So they recruited people between 18 and 22, many of which were the offspring of the original recruits in the study. So it's called the offspring study. And they, of course, now diversified it on a sex base, like men and women, and they even tried to throw in some ethnicity, which was hard in Framingham. So there were over 6,000 participants when this first started. Now there's over 2,500 that still come back every couple of years for this unbelievable battery of tests that they throw at them. They do everything you can think of to these people, and these people come with a smile for 2 days of all-day testing. They'll do it all. They'll run on treadmills. They'll let you do CT scans. You name it, they've done it. They've done everything you can, but what really means is we have reams and reams of data about these patients. Like, they started healthy, now they're old, and they've got all the problems that happen with us with aging and because of your genetics or your lifestyle. But most importantly, we've sequenced them. We know everything about their genome. So now what we said was, what if we could get these patients, get their blood, and make stem cells? So we've done that, and we've done that for hundreds of those individuals now. In fact, the number's upwards of the 700s. And we can try to do some experiments to try to say, could you use stem cells to understand something about common genetic disease? And we weren't ambitious initially. We did the experiment where we were like, could we figure out that sortilin thing that we figured out over 7 or 8 years just using stem cells? And people now make fun of me because they're like, well, Chad, why would you do something that might seem so obvious? I'm here to tell you that 10 years ago when we thought of this, it was not obvious. Like, in fact, we spent millions of NIH dollars, and I had in the back of my mind the possibility this wasn't going to work, right? At the end of the day, I was going to do it, have the result, and it would be like, can't use the cells to do that. That didn't work at all. Now in hindsight, again, it's like, oh yeah, of course, that's obvious, it worked. So we recruited a bunch of patients, we made a bunch of iPS cell lines, and we turned those iPS cell lines into fat and liver cells, just like taking the same tissue from the individuals that had shown up with genotypes. And then we asked, could we learn something similar? This is just some data to show that we could make adipocytes, and this is some data to show that in adipocytes you saw absolutely no change with sortilin. I remember when I talked to the informatics people I was working with after doing the RNA sequencing, they were like, we have thrown everything we've got at this, Chad, and we don't see anything. And I was like, whew. The beauty of blinding it is they had done everything they could to try to show a change, and nothing changed. Like, they were like, there's nothing different. I was like, that's awesome. Then we could make hepatocytes. And in our hepatocytes, we now saw a change. Now, granted, this change is relatively mild, but remember that change was mild to begin with when we first identified it in the individuals. And this magnitude of change looks just like the change you'd expect from GTEx samples that we've looked at across that people have collected. So we now know by RNA sequencing that's probably what you're going to see, and that change is consistent. All 3 of the genes that we would expect to see change in the locus all change according to genotype at a magnitude that's about what we would expect. Interestingly, though, things that we didn't expect happened. Even though there's a very mild change in terms of transcriptional level and the transcripts that are made, there's a huge change in the lipids that these cells now have in fat. So you've got a magnitude order fold change in the way in which they're handling, creating, and storing and issuing lipids. And this is just a very sophisticated mass spec analysis of the lipid composition of these cells, and you can basically see that the triglycerides, the DAGs, a lot of the storage lipids, and even some of the phospholipids are significantly altered just by the alteration of the sortilin gene and probably some of the other machinery involved in lipid handling. There's some other amino acids that we don't totally understand that are changed. And because we probably isolated these cells and they didn't have the same confounding effects, even with a smaller group of cells than the original genetic studies, we were able to get new information about other genes that might be affected by this transcription factor binding site, such as PELTP. Yes?
So just out of curiosity, were you able to look at sortilin levels in the IPS red hepatocytes by Western blot. I mean, so is there a magnitude difference?
Yes, and it's in the public. I usually simplify what is like a remarkable, these days everybody who publishes papers knows you have like 15 supplemental figures and all the, so we have a supplemental figure that shows a Western blot of sortilin, and yes, you can see a protein level difference, you can see an expression level difference, and it's one of these ones where, back in my PhD we used to say if you have to squint it's not worth it. I still have to squint, but it's probably worth it. It's definitely, the order of magnitude makes a difference in human health, and we know that it's probably the right gene. But what stunned me was I learned more about this biology than I had in mice and all the other things we'd done just by doing this experiment. So the conclusion, which to you doesn't seem stunning, but to me was really good, is that you can use these to model common genetic disorders. So voila, we can do things. So there are some other things we learned. There's the variation. So the dirty little secret in iPS cells is that if I make 10 different iPS cells and turn them into hepatocytes, I get 10 slightly different answers because there's a lot of variation. Most of that variation is actually driven by the genetic differences between people or between the cell lines, and not because of the reprogramming or the things you do. That was one of the things we learned and published. We also know that environment matters. We needed far fewer iPS cell lines to find the same thing they found in genetic studies than in genetic studies, and that's probably because of the buffering of environment behavior and everything else that happens on a larger scale in the human population. And then we do believe this is true, and we're doing it today with slightly more sophisticated methods. You can now do real discovery. So rather than rediscovering something I knew already, I can now say, well, what about EQTLs? Now that I have this as a firm basis that it works, I can now go and launch into the discovery mode. And that's what we're doing. And it's pretty simple to see how you might do it. Make a population of these iPS cells, turn them into any cell type you want, especially ones you can't get easily, like aortic vascular smooth muscle cells or the cells in the brain, et cetera, and do these EQTL or even more sophisticated studies, expression studies, splicing studies, to find out what is different. Okay, I'm going to quickly accelerate into the last little bits of the talk, and one of the ones that I'm most excited about, which we haven't published, is this one, and I might skip the next one just to save some time, but this one I'm super excited about, and that is the real dream of this wasn't for me to go to the Framingham study, to these really well-studied individuals and learn something more about them. The real dream was a patient walks through the door at the hospital and I'm like, wow, they've got something wrong. Could I figure out something about them? That was what we tried to launch into here. Can we use iPS cells to understand patient phenotypes? I don't know anything about their genotype. I don't know anything. I just know that they've got a phenotype. And of course, I gamed the system in this one. I went ahead and chose sort of extreme-ish phenotypes, but still, it's just a patient phenotype that I'm starting with. And the patient phenotype I started with in this particular case was people who had diabetes for a really long time, but for some reason didn't have heart attacks, right? And when I first heard about this from cardiologists and endocrinologists, I was like, this just can't happen. Like, I was like, that's probably almost impossible. And I thought, when I said impossible, the bet I made, and I lost a six-pack of Coke on this, was that when you looked in there, you'd probably see atherosclerosis in their coronary artery, and the difference between heart attack was probably like stable plaque versus unstable plaque, et cetera. But these guys were like, no, no, I promise you. And in fact, it turns out it's true. We looked through the patient data of hundreds of individuals at MGH that had diabetes and or heart attack. We just combed through it. And we found patients who had no coronary artery disease, and that's by catheterization. And so I asked myself, why did they catheterize them? I think they catheterized everybody in the 90s. I think that's just what you did. But these people were whistle clean. They, you could buy cath. We looked in there, and there was not a single plaque anywhere to be seen. But they had, in some cases, 30-year course of diabetes, and it wasn't well-controlled diabetes. These are people who had kidney transplants, eye damage, everything you'd expect is a disaster of a complication, but no heart disease. How could that be? The other people are just like you'd expect. They had cardiovascular disease as the major complication of diabetes, sometimes quite quickly in the progression of their diabetes. So we took these, two different populations and a control population, made iPS cells from them, and we didn't even get that many patients, mostly because it's really hard to identify, at least at the way we did, how, as cleanly as we sort of chose, patients with well-established diabetes but no CVD. So we really only found 6 in the patient database, and only 4 of them would sign up for our study. So we made iPS cells. We made endothelial cells and vascular smooth muscle, because my hypothesis was there's maybe something different about the coronary arteries of these individuals that makes them protected versus the coronary arteries of the people who get CAD and eventual heart attack. And then we just looked very simply at transcriptional, what are the differences in the genes? And The first thing that came back was the work that we did on endothelial cells. And I've got like 5 different ways in which bioinformaticians have looked at this. The bottom line is we didn't see anything that was really different, statistically significantly different between one population of endothelial cells and the other population. So I was like, chalk it up, we're just going to lose, this isn't going to work. But then we looked in the vascular smooth muscles. And in this one, finally, we saw things that were statistically significantly different. And interestingly, some of the things we saw were already associated with heart disease. And my favorite one here is down-regulated to the greatest extent was the matrix metalloprotease 13. This MMP13 has already been established by people at the Brigham and Women's to be involved when it's expressed at high levels with a risk for coronary artery disease. So reducing it in mouse models is shown to protect against it. So we already found that as one of the things that might actually, be something you'd be interested in. On the other side, we saw some genes that were the most highly expressed in those people that were protected, but they didn't have any known association with coronary artery disease, but we were interested in it. So this is sort of your top 5 of both of them. A couple of the others we've now figured out are somewhat linked, but on the the overexpressed, we didn't have anything that definitively linked them. As I mentioned, you can go and look at a variety of experiments, and in fact, the professor who did this is what I love about Harvard. You'll read something in the literature and you're like, that's really good. I got to go talk to that guy. And I'm like, oh, he's upstairs. So Matsunori Okawa is actually upstairs. He did most of the work on this gene, and he's definitively linked it to coronary artery disease. So this one, for us at least, was less interesting to study. More interesting to study was this, AADAC, which is thought to be a critical enzyme in transferring triacylglycerides to probably diacylglycerol, so at the ER membrane. So most people think about storage lipids in these like storage vesicles. Most lipid handling in most cells happens at the ER, and this is one of these ER enzymes that's incredibly important in the hydrolysis of lipids. And so we were wondering, like, what the heck is this thing doing? Well, if you look at a textbook, which I have already told you are usually the wrong thing to do when you want to understand biology, but a textbook that tells you what's going to happen in heart disease says the following things happen. You get something that goes wrong here in the arterial wall that leads to this invasion of, there's an inflammatory process that leads to the invasion of immune cells. Those immune cells start to try to protect this thing against the insult, which is typically oxidized LDL, et cetera. They collect more and more of that LDL until they make some sort of plaque or this foam cell, and then you get this big, gigantic atheroma, which eventually will rupture and cause the heart attack. We knew that MP13 worked right here. It's actually essential for the chewing up of the matrix that holds these cells in place, especially vascular smooth muscle cells, that allows them to get involved in this process. Another thing that was a nice clue was just as we were making this discovery, 2 groups found out that contrary to popular belief, where we thought that all of that atherosclerotic lesion was from the immune cells gobbling up the lipid, it turns out 50% or more of that is actually vascular smooth muscle cells that you don't know they're vascular smooth muscle cells because they don't look like them anymore, but that's what they started as. So this is a very important step. Also important is it looks like they start to handle lipid in a different way. They become dysfunctional in their lipid handling, and that's probably what leads to this big fatty plaque. And we wondered, is AADAC involved there? And so this is what we saw in the patient data, that we saw more AADC expressed than people without CVD. So we asked, in vascular smooth muscle cells that we've differentiated, could we turn that up? So we made this DOX-inducible system that would turn it up to the exact same extent that we were seeing in the patients. And now we could ask some questions. What happens when you turn it up in vascular smooth muscle cells? Well, one of the things that happens is you get a reduction in storage lipids. And we've now done that same sophisticated metabolomics on it. And in fact, what it looks like is you get a shift, and I didn't even know this. It's what's beautiful about biology is you get to learn things. And I was like, Kennedy pathway? What's the Kennedy pathway? Well, if I would have been around in 1968, that was the big discovery, how you use lipids to make phospholipids. Is a portion of a biosynthetic pathway that you can actually run either way. And it turns out that this enzyme is probably shifting the Kennedy pathway to catabolize storage lipids to create more phospholipids, which is actually beneficial in heart disease. And we see the changes in the numbers of lipid droplets. You also get changes in the migration of these cells because of the changes in lipids. The cells that are expressing high levels of AADC are less migratory, and we already know that that enhanced migratory process is involved in coronary artery disease. We also see changes in cell proliferation, which is often linked to that, as well as cell death. Of course, we weren't going to stop there. We wanted to know, could this be definitively linked? So we made a mouse. And the way we made this mouse is we actually knocked into the ApoE background, which is one of the few backgrounds that gets atherosclerosis. And my mom used to ask me, like, why do you study things in mouse? And truly, I don't know. It's like the worst model. They don't get heart attacks naturally. We have to do a lot of genetic tricks to make them even look like they're going to have a heart attack, and they still don't get heart attacks. They just get plaques. So the ApoE model actually gets plaques if you don't do anything to it. And if you feed it a high-fat Western diet, it really gets a lot of plaques. So we made a mouse that could overexpress the ADAC specifically in vascular smooth muscle cells on that ApoE background using the CRISPR-Cas system. And this is all just proof that we did that. And when we did that, what's interesting is in those vascular smooth muscle cells that were taken from the mouse, primary, you can see the same reduction in lipid droplets. So they're storing the same storage lipids less. They have the same phenotype in cell migration, same proliferation phenotype, but most importantly, they've got a reduction in atherosclerotic lesions. They're protected against lesions because of the changes we've made. And so our hypothesis right now is that there's something about this ER-specific enzyme that actually protects against excess lipid damage, which is the primary dysfunction in those cells that lead to the plaque, which is now serving as a protective mechanism in individuals who are able to do that. To me, this is stunning, that we could take a relatively rare population of patients that I knew nothing except this interesting phenotype about and get down to potential molecular mechanism for how it might be working to protect you in heart disease. Okay, so I'm going to skip a lot of this. This was the answer to my mother, which is, what do you do with an inexhaustible supply of adipocytes? You can do a small molecule screen. So we did that. We teamed up with a group at Roche. We did a small molecule screen, and we'll just sort of skip ahead to the final figure, which is we figured out how to turn white fat into brown fat by throwing some small molecules on it, which might be a beneficial thing. The problem I find with this model is that when you make brown fat, it's more energetically, it basically uses more energy. And we already know from studies in obesity that people who exercise more eat more. So if I turned on more brown fat in somebody, they'd probably just eat more. So it might not work as a weight loss effect, but it may do a lot of things beneficially metabolically. So I do think that this has now established this as a system by which you can do drug screening and really identify drugs, and it's gotten a lot of large pharma companies interested, and they're more interested in the idea that you can do what I did with Framingham, get a population of people, use those to do the drug screen, and in fact you might call it a sort of phase 0.5 clinical trial. So rather than, here's another fact I didn't even know until Mark Fishman came to our department, and he was the former head of Novartis. He had taught me the following, which was, when's the first time most drugs see patients? Does anybody know? You know, in this long process, billion-dollar process that pharma companies use to, you know, and even not even patients, a human cell, when they're taken by a patient in a phase 1 clinical trial. 99% of drugs never even get tested on a human cell until they're taken by a patient. So you could say that the drug industry might be a little bit broken in its drug discovery paradigm. And we think that this is one way to fix that. Why not use the human cell that you want to change in the first sort of phase of the discovery and then retest it in your sort of safety and the rest? Because you can now make hepatocytes, cardiomyocytes, look at the tox issues of the drug that you're testing in a much more efficient and cost-effective way than you would with patients. And then go into patients, and you might even know patients of a particular genotype are the most, going to benefit the most. Okay. I'm going to, by necessity, talk about some future directions. We're still trying to understand why it is that when you get fat, you die of a heart attack. What is it about being fat that leads to diabetes? And then the ultimate question is, why do we get fat in the first place? It turns out that getting fat in the first place has nothing to do with fat tissue, for the most part, which I can talk about. And we use the same tools. We just sort of turn the crank. The only difference is over the 10 years, we now can make almost every cell type that you're interested in understanding in metabolic disease. We routinely make hepatocyte-like cells, adipocytes, skeletal muscle cells, endothelial cells, pancreatic beta cells, and now the newest cell type we're really playing with are one of the cell types involved in regulating hunger and satiety in your brain. And I'm just going to sort of run through this. Why do you usually die of a heart attack? What can be done about dyslipidemia? Here's the daunting list that faces us. After looking at 300,000 people, we have hundreds of genes that are associated with lipid changes in individuals and linked to heart disease. So as a simple scientist, you're like, how am I going to do anything with it? Well, again, these stem cell models are really powerful. By using CRISPR-Cas gene knockout technology in our hepatocyte-like cells, we set up high-throughput screens, and we've now marched through half of the genes associated and asked, like, what can they do in these high-throughput assays? And you can see one here. The positive control was PCSK9. Those in the know, PCSK9 is the new hot gene. There's an antibody directed against it that lowers cholesterol levels, LDL cholesterol, to the lowest levels possible. When you knock it out, you get about a 30% change, which is consistent with what we find in the individuals. If you knock out ApoB, which carries LDL cholesterol, you completely eliminate it. One of the genes we found is twice as strong as PCSK9, A1CF, which actually is this RNA chaperone protein. And it's not just an RNA chaperone for ApoB, which is what it was originally identified. It's an RNA chaperone for a whole class of lipid regulators. So it almost functions like a microRNA in establishing sort of a lipid biosynthesis pathway. You can see that it's got beneficial effects in total cholesterol, your triglycerides, and your LDL uptake. It's got directional changes in all the right ways. And we figured out that it does that by regulating a very precise gene transcription cascade by chaperoning these various RNAs at the sort of translational level. And I think that that's all I was going to say today. So I wanted to just thank the people that were involved in the work. A lot of the work that I've been doing has been a very close collaboration with the geneticists who spearheaded Chris O'Donnell at the Framingham Heart Study, State Catherayson at MGH. I've started to dig into the metabolic underpinnings. It's not all genes. Sometimes it's changes in metabolites that really are the root cause here, and that's been principally done with Rob Gerson. A lot of the informatics has been spearheaded by a former postdoc, Rahul Deho, who's out at UCSF now, and then a number of critical individuals who have either made the differentiation protocols, the protocols for sort of doing the high-throughput assays, et cetera, that over the years have contributed and now have gone on to bigger and better things. I'd like to thank you all for your attention and be happy to take any questions. Yes?
For the SORT1 study, were you able to identify the transcription factor that may interact with that minor allele and drive expression?
We don't know the specific CBP family member, and part of the problem with that is there's 3 that are highly expressed in liver cells, but it's certainly a CBP family member. We've done the CHIP experiments, and we can show that either in overexpression or in normal hepatocytes, you'll find increased amounts of CBP factors at that site. And it's probably, the dirty little secret is, if I just turn up CBP in a non-hepatocyte cell type, it doesn't work. So it's probably a combination of transcription factors in addition to CBP that are hepatocyte specific that allow the whole transcriptional cascade to kick off. And CBP may just be one of them. CBP, you probably know, has been shown to be one of the critical factors in like DNA looping. So it's probably bringing some other transcriptional regulator nearby.
I may have missed it, but did you make the isogenic cell line?
Oh, I did. And that's a great question. Thanks for asking it. I often put it in the talk, but I tried to cover too much. In the study we did I remember I first showed the sortilin stuff. I trained at UT Southwestern, and I go back and I talk to Mike and Joe Goldstein. Joe's like, you didn't do the right experiment. I'm like, okay, Joe, I know I didn't do the right experiment. He was like, you have to do the Koch's postulate of genetics, which is put the change into the otherwise normal. And so we did that. We made, with gene editing, a minor or a major, and we asked, what's the change there just on that background? And it's exactly what we'd expect. If you make a major a minor, you get an increase in sortilin. If you make a minor a major, you get a decrease. So it's definitely that SNP that's the causal SNP.
So on the AKT2 story, why left side overgrowth?
Ah, great question.
Where did you take your fibroblasts from?
Yeah, so we never got fibroblasts. We ended up doing gene editing to create our own model.
Oh,
yeah, Robert Simple, after 4 years of dickering between the UK and the Harvard with IRBs, and we just gave up. It's impossible. So we just made them ourselves. We don't know why there's left side overgrowth, but one of the most compelling hypotheses that I really like is, I was trained as a developmental biologist, and probably many people know this, that as the node extends in the early developmental process, it has a bunch of flagella that are beating. And interestingly, they're beating directionally. And they're beating directionally in a way that actually determines a lot of left-right asymmetry in individuals. That's why certain things are on different positions. And it's thought, this is known, AKT is known to be in that pathway for flagellar motor function, so it provides some of the energy. So it's possible that you're changing flagellar motion, and that's leading to some overgrowth on a particular side due to early growth factor changes, deposition. But I've never tested it, nobody's tested it, but I always like these attractive hypotheses because at least they're testable, but I've never done it.
Any other questions? No, thank you.